retinal diseases. All lectures will be given by world-renowned retinal experts. We will start with longer duration treatments in AMD by Tian Wong from Singapore. Then we'll move on to longer duration treatments in diabetic macular edema by Diane Doe from United States. The topic coming up management options in geographic atrophy will be presented by Professor Ashad Kanani from United States. Addressing safety issues with long-term treatments, this talk will be given by Professor Caroline Bomal from United States and Professor Tund Pito from Ireland. We'll talk about functional and morphological methods for long-range patients monitoring. And finally, Professor Hendrik School from Switzerland will discuss emerging therapies for Stargardt disease. At the end, there will be time for discussion. You can send your questions via chat. Me and Professor Paolo Lancetta, we are looking for you to have an exciting session.
Hi, I'm Christina Wang. I'm on faculty at the Baylor College of Medicine, and today I'll be speaking on longer duration treatments for wet AMD, a topic that's top of mind for many of us. Thank you to the WOC meeting organizers, and particularly to my dear friend, Dr. Anat Lowenstein, for the invitation to be part of this year's Congress. These are my disclosures with pertinent entities underlined. We are very fortunate in 2022 to have not just one, but several very effective treatments for wet AMD. That being said, in order for that effectiveness to be sustained, these anti-VEGF agents often need to be given regularly and frequently, demanding a treatment burden that many patients are unable to endure long-term. Compared to our pivotal trials, real-world patients are relatively undertreated. You can see from the bar graph there that they also, in turn, have much more modest visual acuity gains. The heavy treatment burden required of patients was also shown in the TREX AMD study, which showed that the average patient could not extend beyond 8.4 weeks, with almost a quarter of them requiring monthly injections in order to control disease activity. So that brings us to present day and why there is such a quest for durability. While this list is not comprehensive, the landscape of wet AMD therapeutics is ripe with innovation. Starting on the left, we have great agents today, including faricimab, the port delivery system, and 2 biosimilars, all of which were just approved within the last year. Moving to the right, we have next-generation anti-VEGF agents that target not just VEGF-A, but also VEGF-C and D. Below that are several gene therapies. And then to the right of the slide, there are drugs looking at different mechanisms of action, including tyrosine kinase inhibitors. Since we obviously can't cover all of these in 8 minutes, I'd like to focus on the ones highlighted and how they may affect our patients and offer them the longer durability that we're all seeking. Let's start off by briefly talking about 2 therapeutics that have only recently become commercially available. Faricimab works by targeting both VEGF-A and angiopoietin-2, which acts on the Tie2 pathway. The phase 3 trials, Tenaya and Lucerne, compared faricimab every 8, 12, and 16 weeks with aflibercept every 8 weeks and found comparable visual and anatomic outcomes between the 2 groups. But the advantage of faricimab seemed to be in its durability, with over 60% of the faricimab group achieving every 16-week dosing and about 80% achieving 12-plus week dosing at the 2-year time point. The port delivery system with ranibizumab was studied in the phase 3 Archway study and has shown visual and anatomic stability for over 3 years now compared to conventional monthly ranibizumab. Impressively, 95% of patients did not require supplemental therapy before the mandated Q6-month refill exchange. And in the phase 2 ladder study, where patients were allowed to go even longer, the median time to refill was 15.8 months in the highest dose group. Now, of course, there are unique risks associated with this surgically implanted device, but many of these can likely be mitigated with proper technique and experience. Next, let's take a look at some of the agents targeting VEGF. First, a drug familiar to all of us, aflibercept, is being evaluated in a high-dose form that is 4 times the standard dose in a slightly larger 0.07 cc volume. The phase 2 Candela study suggested that there might be slightly better anatomic outcomes with high dose versus standard aflibercept captured in the graph to the right, and help support the development of a phase 3 program which is looking to treat as infrequently as every 16 weeks. KSI-301 is a molecule that blocks all VEGF-A isoforms, but what's unique about it is that it's mounted on an antibody biopolymer conjugate platform with several beneficial properties. KSI-301 was striving for a really high bar of every 20-week treatment, and so the phase 3 study was designed to compare KSI-301 every 12, 16, and 20 weeks to aflibercept every 8 weeks. However, DAZZLE did not meet its primary endpoint of non-inferior visual acuity outcomes, and as you can see from the graph, this was likely due to undertreatment driven by the Q12-week group. I think we learned some really important lessons from the DAZZLE study, namely, no matter how durable any drug is, there will always be a subset of patients who require more frequent treatment. Now let's look at one of the categories I'm really excited about, gene therapy. When we talk about longer durability therapeutics, truly no class of treatment sets a higher potential bar than gene therapy. Farthest along the developmental pipeline is RGX-314, an AAV8-based gene therapy that encodes for an antibody fragment similar to ranibizumab. The swim lane plot below illustrates 3 highest dose cohorts where each dot represents an anti-VEGF injection. You can see that these patients required very frequent treatment before RGX-314 was administered, but most were able to significantly decrease their injection burden afterwards. And in fact, there was a 79% decrease in annualized treatment burden in cohort 5 by the end of 2 years. Subretinal RGX-314 is being examined in 2 pivotal trials, Atmosphere and Ascent, and a suprachoroidal approach is also being evaluated in a phase 2 study called AV8. Next, let's move on to the tyrosine kinase inhibitors, or TKIs, which have a broader mechanism of action compared to targeted anti-VEGFs and are anticipated to offer durability somewhere in the range of 6 to 9 months. This group of treatment works from an intracellular standpoint and ultimately blocks signaling of VEGF receptors 1, 2, 3, and the PDGF receptor. Several TKIs are in the pipeline, including OTX-TKI, which is a polyethylene glycol hydrogel-based biodegradable intravitreal implant that releases axitinib. The phase 1 study in Australia studied 4 cohorts of patients with wet AMD. And as you can see from the chart, over 60% were able to go 6 or more months before requiring rescue treatment. There is currently a phase 1b study based out of the United States that is ongoing, for which we will likely hear results very soon. EYP-1901 is another TKI, vorolanib, that is an intravitreal injection of a bioerodible insert. The phase 1 DAVIO study included patients with wet AMD and showed a 74% reduction in treatment burden at 12 months, with over half of patients being supplemental injection free at the 6-month time point. A phase 2 trial is expected to launch soon. And last but not least, I want to spend a moment talking about this catch-all category. AKST-4290 is a CCR3 inhibitor in the form of an oral pill. ICON-1 is a modified factor 7A protein injected intravitreally, which binds tissue factor. AXT-107 is an intravitreal gel depot that inhibits VEGF-A and VEGF-C and activates Tie2. And UBX1325 is an intravitreally injected senolytic molecule. The rationale behind this is that if too numerous, aged senescent cells can actually compromise vascular integrity, which contributes to the pathogenesis of wet AMD. By inhibiting BCL-XL, which senescent cells depend on for survival, UBX1325 actually promotes apoptosis of these cells, restoring tissue health. The phase 1 study confirmed that UBX1325 was well tolerated and able to maintain visual stability for 6 months. The phase 2 ENVISION study will load with 2 injections and then follow patients over 6 months. Interim data should be presented before the end of the year. So I hope that gave you a taste of all the exciting candidates that may offer longer durability in the treatment of wet AMD. By addressing the undertreatment, which goes hand in hand with treatment burden, there's a real opportunity to continue improving visual outcomes for our patients in the near future. Feel free to reach out with any questions, and thank you again to the WOC for the invitation to present.
Hi, my name is Dr. Diana Doe, Professor of Ophthalmology and Vice Chair of Clinical Affairs at the Byers Eye Institute of Stanford University. It's my pleasure to provide an update on diabetic macular edema treatments. My disclosures are noted here. What happens when we treat patients with diabetic macular edema? Several retrospective studies have shown that real-world outcomes for DME are far inferior to the ones we see in clinical trials. Why is this happening? Most likely, undertreatment is the main culprit why patients in our clinics do not achieve the excellent visual acuity outcomes seen in pivotal clinical trials. Because this is an unmet need, many new medicines being developed for retinal vascular diseases and diabetic macular edema are looking to provide more sustained VEGF inhibition. One of these new agents, faricimab, inhibits both VEGF and angiopoietin-2. Faricimab was recently FDA approved in the United States for the treatment of diabetic macular edema. Now let's review some of the data from the pivotal Yosemite and Rhine clinical trials, which evaluated 6 milligram dosing of faricimab compared to aflibercept. In this clinical trial, the faricimab-treated patients were on a personalized treatment interval algorithm, which was driven by treat and extend regimen with decisions based on changes in OCT and visual acuity. The primary outcome of the clinical trials was at year 1, and faricimab was non-inferior to aflibercept. Moreover, at the year 2 follow-up visit, we could see that the vision gains with faricimab, when it was administered every 8 weeks or up to every 16 weeks based on the personalized treatment interval, were not inferior to aflibercept given every 8 weeks on label. Furthermore, more than 50% of patients in the faricimab personalized treatment arms achieved every 16-week dosing at week 52. If we look at what happens at week 96, we can see the proportion of patients who achieved 16-week dosing increased to 60% at year 2. What happens to the anatomic response? We also see through 2 years of follow-up that faricimab provided very rapid and sustained reductions in retinal thickness compared to aflibercept. The safety of faricimab was also well tolerated, with the intraocular inflammation events quite low, just slightly higher than the aflibercept treated arms, but there were no cases of retinal vasculitis or occlusive retinal disease. Another agent that is in pivotal clinical trials is KSI-301, a unique antibody biopolymer conjugate that inhibits VEGF. The phase 1b diabetic macular edema clinical trial had follow-up and treatment through 1 year. And when we look at the efficacy of KSI-301 in diabetic macular edema, we could see that after 3 loading doses and then as needed dosing, KSI-301 provided improvement in visual acuity of approximately 7.6 letters with sustained reductions in retinal thickness. This was all achieved with a mean injection of 4 over the 1 year period, and that includes the 3 loading doses. This large phase 1b study suggests there may be durability of KSI-301 in diabetic macular edema, because in this retinal vascular disease, 50% of KSI-treated eyes did not require any retreatment. Of course, a larger pivotal clinical trial is currently underway to truly identify if KSI-301 has durability. What happens with the diabetic retinopathy? We can see that from the phase 1b study that after 3 loading doses and then 2 additional doses through 72 weeks of follow-up, there is impressive regression of the proliferative diabetic retinopathy disease, and this converts to non-proliferative disease over a 72-week follow-up period. In summary, diabetic macular edema treatment options are increasing for our patients. Anti-VEGF therapy remains the gold standard, but new therapies, such as faricimab, provide additional treatment options, which may allow for longer treatment intervals. In addition, new medicines, such as KSI 301 and other medicines in development from other industry partners, also provide other possibilities for our patients with diabetic eye disease. Thank you for allowing me to share this information with you.
I'll be reviewing safety issues with long-duration treatments. These are my disclosures. The last 2 decades have primarily been an anti-VEGF monotherapy era, where frequent injections are required to give optimal visual results. However, this creates a treatment burden on patients, and there's no clear endpoint inducing stress related to therapy. As well, 30% of patients continue to lose vision. For this reason, novel and pipeline agents and delivery systems are being developed for AMD and DME, and I'll be reviewing faricimab, brolucizumab, and the port delivery system and safety. Faricimab is the first bispecific antibody designed for intraocular use. This single molecule has one Fab that binds and neutralizes Ang2 and another Fab that binds and neutralizes VEGF-A. Ang1 is a key player in the angiopoietin Tie2 pathway, and this is important for vascular stability, as in the scenario here where Ang1 is bound to the Tie2 receptor and the VEGF-A receptor is unbound. However, in pathologic stress conditions, there's an angiogenic switch, and Ang2 is upregulated, which causes vascular instability. Ang2 also sensitizes blood vessels to the activity of VEGF-A, promoting angiogenesis and vascular permeability. Faricimab has been evaluated in multiple clinical development programs, the phase 3 Tenaya and Lucerne studies for neovascular AMD, and the phase 3 Yosemite and Rhine studies for diabetic macular edema. In the Yosemite and Rhine studies, patients were evaluated with faricimab fixed every 8-week dosing, or according to a personalized treatment interval where patients could be dosed as infrequently as every 16 weeks after loading doses within a aflibercept comparator arm. Faricimab overall was well tolerated throughout the study with an acceptable safety profile comparable to aflibercept. The incident rates of intraocular inflammation events with faricimab were low and comparable to aflibercept, and there were no cases of retinal vasculitis or occlusive retinal vasculitis reported during the trials. Only a small number of retinal occlusive events were reported across all arms of the study. Over 2 years, faricimab demonstrated durable efficacy up to every 16-week dosing in 62% of eyes, and at every 12-week or greater dosing in 78% of eyes with impressive safety. The Tenaya and Lucerne studies were phase 3 global randomized double-masked 112-week trials designed to assess efficacy, safety, and durability of faricimab. And patients were treated with fixed dosing faricimab every 8, 12, or 16 weeks in year 1 after loading doses, followed by a personalized treatment interval in year 2. As well, in the Tenaya and Lucerne, faricimab was well tolerated with an acceptable safety profile through week 112. Overall, there were no unexpected adverse events in the faricimab or aflibercept neovascular AMD populations treated with these intravitreal injections. And the rates of intraocular inflammation pooled were comparable between the treatment arms and low at 3% in the faricimab versus 2.3% in the aflibercept arm. There were no cases of retinal vasculitis or intraocular inflammation associated with retinal occlusive events reported in either study. The 2-year data demonstrate strong durability with faricimab. 63% of patients were on every 16-week intervals and 70% on every 12-week or greater intervals, and there were no new safety signals, notably no cases of retinal vasculitis or occlusive retinal vasculitis. Switching to a different agent, brolucizumab 6 mg was approved for treatment of neovascular AMD and more recently diabetic macular edema. However, shortly after initial approval, there were noted cases of intraocular inflammation, and this could be associated with retinal vasculitis and/or retinal artery occlusion. Risk factors include bilateral same-day brolucizumab injection, female gender, possibly history of ocular inflammation or retinal vasculitis. This inflammation is often delayed in onset with a possible immune pathogenesis. Brolucizumab is a 26 kilodalton single-chain variable fragment, and due to its smaller size, this allows for higher dosing per volume and a potential longer-lasting effect. This shows the multiple brolucizumab clinical programs, some of which were terminated due to frequent dosing and the potential risk of intraocular inflammation noted with every 4-week dosing. Based on the HAWK and HARRIER phase 3 studies for neovascular AMD, just over 50% of patients were on every 12-week dosing. However, multiple case reports of occlusive retinal vasculitis were published shortly after brolucizumab approval, as demonstrated here. And this lists the variable clinical features that have been reported associated with this retinal occlusive vasculitis. Here is one such case. Note that the arterial narrowing can be subtle and is more visibly seen on fluorescein angiography. Novartis commissioned a safety review committee to evaluate patients in the HAWK and HARRIER, and they found that the rate of intraocular inflammation was 4.6%, intraocular inflammation with retinal vasculitis was 3.3%, and intraocular inflammation with retinal vasculitis and retinal artery occlusion was 2.1% from patients in the trial. And based on this, a diagnosis and treatment algorithm was designed. The last thing to discuss is the port delivery system, which is this implantable device in the operating room filled with customized ranibizumab to allow for continuous intravitreal delivery of a customized formulation of ranibizumab. While this is placed initially in the operating room, it's refilled in the office, and this is an alternative to frequent intravitreal injections. Looking at ocular adverse events, through week 79, these were similar to the primary analysis, and special interest events include conjunctiva, bleb or erosion, endophthalmitis, and implant dislocation. To summarize, new modalities may offer improved durability and better efficacy to optimize outcomes, but it's critical that we have safety with any novel therapeutics for our patients in these high-risk groups, especially of neovascular AMD and diabetic macular edema. Thank you.